Hi, my name is Hannah Green. Today I'm going to talk to you about my PhD research studying carbonate chemistry in the Arctic Ocean using satellite Earth observation. I'm based at Plymouth Marine Labs and the University of Exeter here in the UK, and I'm funded by AXA XL Ocean Risk Scholarships. And this work is also in collaboration with ESA and LIVA. Ocean acidification is a greater threat in the Arctic for a number of reasons. Firstly, the Arctic has a reduced buffering capacity to the absorption of CO2 due to increased freshening from river runoff and ice melt, which makes it particularly susceptible to OA. Recent assessment suggests regions in the Arctic are already seasonally corrosive to aragonite as a result of OA. Warming further increases the Arctic's susceptibility to OA with continued loss of multi-year ice, increasing the surface area available for CO2 gas exchange, while lower salinity and total alkalinity reduces the buffering capacity. The rate of warming in the Arctic is 2 times greater than at lower latitudes. This is caused by a process of positive feedback loops. What this means is organisms are facing multiple stresses, which can affect their ability to adapt and tolerate OA. The Arctic is extremely regionally diverse, so OA will not happen uniformly, which will be discussed in the next slide in more detail. As temperatures warm at lower latitudes, boreal species are moving northwards to maintain their thermotolerance thresholds. As mentioned in the last slide, the Arctic is regionally diverse. Here, the Arctic Ocean has been divided into 5 regions based on previous work by Carbonate and Wasman in 2006 and Finley et al. 2015. Each region of the Arctic is experiencing different stresses as pictured in this figure. The regional diversity of the Arctic affects how each region is currently responding to climate change pressures and how they will respond in the future. Therefore, to understand how climate change is impacting the Arctic Ocean, it needs to be studied from a regional perspective. So our methodology was based on looking at the Arctic regionally. First of all, use the Sea Around Us database to find the top landed species of each Arctic region. A literature search was then carried out on these species to see if any or what impact OA studies had been carried out. Here is a summary of the results. The review identified 3 key knowledge gaps. Knowledge gap 1, lack of studies on OA impacts on fish and their supporting food webs. To date, only 10 of the top 47 landed species in the Arctic have been studied for OA impacts, and not all of these studies were performed on Arctic populations. Responses were found to vary with species and life stage. Though 8 of the 10 species studied showed negative responses to OA in one or several of their life stages, particularly larvae and juveniles. Knowledge gap 2, lack of understanding of multiple stresses. We saw in the last slide that each region is experiencing different combinations of environmental change and stresses. Tolerance capacity of organisms to OA has been shown to differ with additional stresses. It is therefore not possible to understand the impact of OA combined with additional climate change stresses, as studies do not take into consideration all these stresses. Only 19 of the OA impact studies found in literature research had OA impacts combined with another stressor. The main additional stressor tested was temperature, but food was also used as a secondary stressor in several studies. Knowledge gap 3. What environmental conditions do Arctic organisms experience? Regional heterogeneity of the Arctic is largely not represented in experimental studies. Experimental OA impacts on Arctic commercially important species use pH levels in their control treatments, much higher than pH found in some regions of the Arctic. The Arctic Ocean has a large seasonal variability, and organisms are unlikely to experience the stable conditions usually used in laboratory experiments, and are likely to experience different conditions at different life developmental stages. We then recommended a series of steps to address these knowledge gaps. Firstly, establish baselines and continue observation, so identification of when thresholds may be reached, whether this be at the ecosystem or organism level. Understand and monitor the long-term change and variability in carbonate chemistry and other climate change stresses relevant to multiple generations. And thirdly, map fish life histories onto satellite observational products to assess what conditions populations presently spend their time at different life stages, and identify what key stresses are for specific organisms or ecosystems. And lastly, incorporate species baselines into experimental studies. You can see here the chemical reaction that happens when CO2 is absorbed into the water column known as OA. We can monitor this reaction by looking at 4 carbonate system parameters: pH, dissolved inorganic carbon, total alkalinity, and PCO2. If you measure any 2 of these parameters, you can calculate the other 2 from them. So how do we measure carbonate chemistry using satellites? The satellite sensors measure oceanographic variables such as sea surface salinity, sea surface temperature, and ocean colour, which chlorophyll A can be derived from. These can be used to calculate carbonate chemistry parameters using empirical relationships. Empirical relationships were first developed using in situ data. Now satellite data can be used as an input data set. Once we've derived total alkalinity and dissolved inorganic carbon from satellite data, we can then use a carbonate system modelling package to calculate pH. The next step in the research was then to work on establishing these environmental baselines. We went about this by first evaluating the published relationships for total alkalinity and dissolved inorganic carbon using a combination of input data sets shown here. Analysed this using the method outlined in Land et al. 2019. A combination of data sets were inputted into a match of data set. Using the match of data set database as a test validation data set, this data was then divided into the regions identified in my earlier work. A set of relationships predicting dissolved organic carbon and total alkalinity was compared. Optimal relationships were then selected, and these can then be used to calculate the gridded time series of total alkalinity, dissolved organic carbon, and other carbonate system packages. This is an example of the output from the algorithm comparison discussed in the last slide. This figure was produced for the analysis of Atlantic influent seas, where the current published relationships had the highest RMSDE for each input combination. The left-hand side of the graph shows input combinations from different data sets. The right-hand side of the graph shows RMSDE for the input data set with the best performing algorithm. The take-home message from this is for total alkalinity in the Atlantic influent seas, the best input combination was the sea surface salinity ISAS dataset, which stands for in situ sea surface salinity gridded field. However, all inputs, including satellite inputs, had an RMSDE less than 25 micromoles per kilogram. This figure is the output also from the Atlantic influent seas, but for dissolved inorganic carbon, with the results again showing the highest RMSDE for each input combination. In summary, for dissolved inorganic carbon in the Atlantic influent seas, the best input combination for sea surface salinity is the satellite input to the Beck Arctic dataset, which stands for the Barcelona Expert Center dataset for high latitude and Arctic SMOS data. All satellite inputs for salinity outperformed others, and the best sea surface temperature input was also a satellite dataset, the ESA Climate Change Initiative Austria dataset. But there was only a minor difference between other sea surface temperature datasets. Here is an example of the gridded output that can be produced from these algorithms. This animation shows total alkalinity in the Atlantic region using the relationship from Nondor et al. 2009, from April 2015 to the end of 2016. As you can see, it has captured the seasonal changes in total alkalinity nicely throughout the year and a half of animation. Not all relationships are performing as well. The Pacific region had a high RMSDE compared to the Atlantic and outflow shelf regions. You can see this in the figure, in this figure, and in summary, the Pacific influence these relationships need further development. It is the same for dissolved organic carbon relationship in the Pacific region, where the RMSDE are high and need further development. One reason for the higher RMSDE of the Pacific region could be due to the algorithm not capturing upwelling over the shelf break. This figure shows the residuals of total alkalinity. You can see here that there's the largest difference is around the shelf region, which is captured in this figure by the pinky purple and red colours around here. Once the current performance of published algorithms had been analysed, the next step was to improve the performance. Using the Atlantic influence seas as a starting point, the correlation between oceanographic variables and total alkalinity was first analysed. This figure is a correlation heat map of different oceanographic variables. Unsurprisingly, there is a strong correlation between sea surface salinity and total alkalinity. After looking at the correlation with total alkalinity with other oceanographic variables, sea surface salinity had the highest correlation. Here is a plot of total alkalinity and sea surface salinity relationship in the Atlantic influence seas. It's clear from this figure there is two relationships for this region, one below and one above the line. The next step was to find out what factors causing this difference in relationship. In previous work by Gregor et al. 2010 showed a spring-summer split. So the next step was to look at the data seasonally. Here, the data is plotted into 4 seasons, and a regression line is calculated for each season and plotted for all 4 graphs. Spring-summer regression lines are almost the same, but there is a clear difference between that with autumn. In winter, there is so little salinity range, it is hard to make out the relationship. We then looked at spring-autumn relationships more closely. Here's a plot of the absolute residuals of spring-autumn relationship by day of the year. Here you can see the spring relationship fits best between day 100 and 285, and the autumn relationship fits best and has lower residuals the rest of the year. That being said, the next steps in research are to firstly further improve and develop novel regional empirical relationships to reduce their uncertainties, evaluating whether the relationships capture the nuances of the Arctic Ocean, including ice melt and upwelling events. And secondly, exploiting these novel relationships to identify which regions are experiencing highly variable pH and organic saturation. And thirdly, evaluating which areas, ecosystems, and economically important fish species in the Arctic Ocean are most at risk from ocean acidification and sudden low pH upwelling. This will include analysing what this means for specific Arctic fisheries and identifying which species should be studied further where knowledge on their susceptibility to OA is lacking. Thank you for listening and any questions.